Let's do that again. Shh. Attach earlobe. Okay, let's get started. Today we're going to talk about part 2 of the genetics. So hopefully what you learned last time is going to help you today. Let's see. So last time you learned about the 3 principles of inheritance. First one is segregation. So this is a quick review. Segregation is talking about how the homologous chromosomes segregated during meiosis, and each one of those homologous chromosomes will carry a distinct allele that could be different. So the allele gets segregated during meiosis 1, right? Principle of dominance is talking about when 2 alleles are present. If one of those allele always express over the other ones, and that's the dominant allele. Principle of independent assortment is based on meiosis metaphase 1. When the tetra get to the middle of the cells, they will face either the paternal and the maternal chromosomes can face either of the poles randomly. So then you get different assortment of maternal and paternal chromosomes. Okay. Now, but science is always like this. If Mendel has started with human or any other animals instead of peas, he wouldn't have figured out all these principles. So today we're going to talk about the exceptions to Mendel's law. Okay, so let's talk about sex-linked inheritance first. But the 2 other ones we're going to talk about is codominance and incomplete dominance. Now remember, these are exceptions to Mendel's law. Now let's look at this karyotype. You should know when you see a picture like this, it's always a karyotype, and this is a human. If you can see it, let me turn on my laser pointer. For human, we have 23 pairs, so it goes from number 1 all the way to 22. So the first 22 pairs are called autosomes, okay, which means that they have nothing to do with sex. The last pair, the 23rd, they are called sex chromosome because this is the pair of chromosomes that determine whether you're male or female. Okay, so male, of course, is XY, female is XX. So let's try this question. Yep, A is correct. Good, everyone. So you have the first 22 pairs, and then last pair is X and Y. We're going to go increasingly difficult, so don't get too happy yet. So if you look at the sex chromosomes, so this is showing you the germ cell of a male with the X and Y, right? So even X and Y chromosome, they do not look anything alike, but they are considered homologous because those are the ones that determine sex, okay? So if you look at this, these homologous chromosomes also segregate during meiosis 1. So the X chromosome will go to one daughter cell, and the Y chromosome will go to another daughter cell, okay? So at the end, you can see in male gametes, 50% will have the X chromosome and 50% will have the Y. But in female gamete, so let's look at if this is female, we'll have two eggs looking like this next to each other. And then so every one of our gametes will contain X. We have no Y. Good, we just talked about this. So 100% of women's eggs will contain X chromosome. Let's try this. Wow, everybody vote for B. I don't even have to explain. Perfect. B is correct because there's only one X chromosome. So if anything, any allele or genes is carried on X chromosome, man will only have one copy of those. He won't have two. Female will have two copies because we have two X chromosomes. Yep, correct is B. Once again, if you need two copies to be heterozygous for something, right? So remember this. So for all the first 22 pairs, for all the autosomes, man can be heterozygous if the genes are carried on any of those autosomes. But if the genes are carried on sex chromosomes, especially on X chromosomes, man cannot be heterozygous because they don't have two X again. Now let's talk about sex-linked inheritance. What I mean by that is that the genetic characteristic that we are tracking is linked to sex, whether male or female, okay? So here's a quick rundown. So female, we have two eggs. So we can be homozygous or heterozygous for any genes that's carried on the X chromosome. Make sense? Men, on the other hand, only have one X because it's XY. So for any genes that's carried on the X chromosome, they are called hemizygous. Hemi means half. They only have one. That's why it's hemizygous. They cannot be heterozygous. They cannot be homozygous. They can only be hemizygous. So any genes that's located on either the X or the Y chromosome is called sex-linked. Okay, some of the genes is on X chromosome is X-linked. If the gene is located on the Y chromosome, it's called Y-linked. Okay, genes that are located in the autosome, those are not sex-linked. Okay, we're going to go into detail what sex-linked means. That means certain disease or the genetic characteristic are only occurring or mostly occurring in female or in male. Okay. Don't pick C, either A or B. So we have mostly B, and why is it mostly B? So B is incorrect. A is correct. Remember, man only has one X chromosome. So recessive X-linked trait means that the recessive gene is on the X. Since there's no other X, so the man has to express that recessive gene if it's located on X. So the answer is A. Now that is explained in this slide here. Because woman has two eggs, so in order for woman to express any recessive genes on X chromosomes, we have to be homozygous recessive, like what we talked about last time. Two lowercase, right? So then we can, because it's nothing dominant, so we will be able to see the recessive gene. But for man, since there's only one X, if he inherit the recessive X-linked characteristic on the X chromosome, then he will have to express it because there's no other X right next to this. Let's say the recessive one is on this one. There's no other X there to compensate or to dominate over it. So there's hemizygous. That's why when they have recessive genes on X, men always have to express it. Now that explains why genetically male is actually more fragile because they don't have two eggs. So if anything's wrong with that one egg that he got, he's done for. For example, all these X-linked disease we're going to talk about, especially the first one, is specific or mostly occurring to male because of that one X or the hemizygote genotype. Okay, so for example, hemophilia is a blood clotting disease that if you get a cut, if the patient get a cut, they cannot form that. It will die from bleeding excessively. Okay, then you have the red-green color blindness. That explains why a lot of men are colorblind and fewer females are colorblind, because once again, you only need one bad X to show this. And muscular dystrophy is another one. Now here's an example showing you why these diseases are more prominent in men. Now, so if you have, so hemophilia is an X-linked, that means the gene is located on X chromosomes, right? So you usually have a father that looks normal, so normal X, normal Y. But the mother can be a carrier, means that she looked perfectly normal. She has no disease whatsoever, but she carried one of the bad gene in X. For a female to show this particular disease, both of these X has to be bad. So she need to be a homozygous recessive, okay? But when female is a heterozygous like this per person here, she's actually a carrier without any symptoms. So no one will ever know that she's carrying a bad gene. Okay. And then when they have babies, if you look at this, the daughter will get one X from the father, right? Get one X from the mother. So this girl get the good X from the father and the good X from the mother, and she's perfectly good. On the other hand, the other daughter can get the good X from the father, but one of the bad X. But she is still a good normal person because she's a carrier. No symptoms is shown. On the other hand, remember the son has to get the Y from the father, right? So the Y is from the father, and the X has to be from the mother. So the son can either get the normal X or the bad X. So this son is good, get the normal X chromosomes, and this son will get the bad one. Can you see that how the son will have a higher chance of getting the disease because all they need is one bad X, but the female will need two homozygous, and it's harder to get two bad ones together than just one in the son. So in this scenario, 50% of the son will have this hemophilia. Probably right, because the baldness gene, well, baldness has a lot to do with environmental factors too. But the gene, it is carried in X. So if the man gets a bad X chromosome from the female, they have a higher tendency to go bald. But hormone plays a role and all that stuff too. Now, when you work on sex-linked genetic questions like this, what you need to do, I'm going to give you an example of this one. A man with hemophilia. So a man is always X, Y, right? And he has hemophilia. So hemophilia gene is carried on the X. Remember, I just showed you the example before. So if he has hemophilia, hemophilia is recessive. So I'm going to put a small H here. Okay. He's going to marry, or he marries a woman that is a carrier. So a woman look like this, 2X, correct? And she's a carrier. That means that one of the allele is normal, big H, dominant, and the other allele is recessive. Okay, so you can see this woman is a carrier and she has no symptoms, but this man has hemophilia. So if you do the cross with Putnam square, so when you do sex link, you always have to draw XY and XX of the parents. So then when you do this, you need to segregate. So for this man, his gamete will look like this, right? 50% will be X, the other 50% will be Y. For the female, 50% will have the normal gene, 50% will have the recessive gene like this. Then you do your little square. Now, they're asking about daughters. So when you read genetic questions, you need to be very careful. Are they asking about all the kids? Or are they just asking about sons? Or are they just asking about daughters? You know that there's 50% chance to get daughters. So out of those 50% chance of daughters, how many? So out of those $2, right? You get 4 kids. Of those $2, how many of those daughters will have this problem, right? So that's how you're going to do it. So if you finish this Putnam square, and you will see what I'm trying to say. So if you do this, you have 2 big X here. This is a daughter, right? That is a carrier. Have a good H and a small H. This one will also be a daughter, but this daughter will be having the problem. This will be a sun. That is normal, right? This is going to be the sun. That is not normal. So you see, out of the $2, one of the two gets it. So you're right. Answer is C. Can you see that? But if I change the question and say, what is the chance that one of the offspring will have hemophilia? Then you will still say 50% because it's 2 out of 4, right? But you will look at different squares in that way. So I need to be careful about what I'm asking in the genetic questions. So good, 50% is correct. Now, so we talked about the first one already, which is sex-linked, right? So the gene of interest could be on either the X and the Y chromosomes, and because of that, either the female or the male offspring will have the disease that you're looking at. Now, the other exception here, remember in Mendel's law, he talked about the law of segregation. But one exception to the law of segregation is that what if those genes, so they're looking at genes that are on different, remember when we talked about law of segregation, dihybrid cross last time, we're looking at genes that are on different chromosomes, right? Chromosome number 1 versus chromosome number 10. So they will segregate or independent assort randomly, right? But in this case here, what if those genes are very close to each other on the same chromosomes? Okay, so if genes are very close together on the same chromosome, they're called linked. So they're linked together. The reason they're called linked together because they usually don't segregate. They travel together. So that, remember, this is trying to remind you that linkage and sex-linked is different. Linkage just means that those two genes are very close to each other, and they can be on autosomes, right? Sex-linked, we're just specifically referring to the sex chromosomes, okay? And so linked genes here is exception to the principle of independent assortment. So here's an example. These two genes, P and R, are very close to each other, okay? So, so you can see that when there's segregation going on, these two genes always go into the same gametes. Now that explains something. Have you seen in your relatives or friends certain characteristics always go hand in hand? For example, red-haired people seems like always have green eyes. So there's certain characteristics that always occur together, and that could be considered linked because they just travel together. Okay. But here it's talking about linkage. It's not absolute. Means that even the genes are very close together, they sometimes can be segregated, but that only happens when there's crossing over. Okay. So here's an example. Now the yellow gene here and the white one, they're very close to each other. Okay, so they're called linked. But so is the yellow one and this black one here. They are on the same chromosome, but they're considered linked also. Okay. But so the yellow and the white one, the chances of these two guys segregated is very unlikely because they're just so close. Okay. But the yellow and the black one segregate is very can happen because remember there's crossing over. So if this part of the gene cross over with this part, then they will segregate. So crossing over is the only time that linked gene, which is on the same chromosome, can segregate. So the principle here is if the genes are really close together, they're unlikely to segregate. But if genes on the chromosome are farther apart like this, they are more likely to segregate. So we're going to talk about this later on. So when they map genes, they will say the yellow and the white one is like one map unit apart, and that can also be called 1%. The 1% means that they have 1% chance of crossing over because they're so close together. The chances of crossing over is unlikely. but the map unit, the distance between the yellow and the black, let's say it's 20 map unit, and that also says that it's 20% chance they can cross over because there's so much distance between them, okay, even though they're on the same chromosome. So that's how they figure out if they see certain genes cross over a lot on the same chromosome, that implies that that gene and the other gene has to be far apart. Good, good, good. A is correct. Let's look at the next one. Now, so based on the crossing over probability, which is a percentage, you can figure out a genetic map between genes, okay? So here's an example. Here's blue is chromosomes here. We have 3 different genes, A, B, and C. So the information given here is that so the chances A segregating from B is 12%. In other words, the chances of A and B separating during meiosis is 12%. So the 12% chance of crossing over could happen. And it's also called 12 map units, okay? So this is the same thing, 12 map units, 12% chance of segregating, 12% chance of crossing over, they're all the same thing. Now, between B and C, there's only 3 map units distance. That means they're closer together. Lower chance of crossing over, 3%. Lower chance of segregating. Can you see that? So let's play with a game. So the main thing to remember about this kind of linkage is that number one, those 2 genes you're looking at has to be on the same chromosome, and the distance between those 2 genes will determine how frequently they get to inherit together. So here's an example. Why don't you guys try to work on it a little bit, and I'll tell you how to do it. So what do you guys think the order of the genes should be? So you should type your answer as the order of the gene. Is it Z, X, Y, Y, Z, X, X, Y, Z? You know, just type it up so I'll see what you have. Okay, so you guys have X, Y, Z, X, Y, Z. Everybody say X, Y, Z. Good. Let's see if it's correct. Okay, so we know that X is 10 map units from the end of the chromosome number 5. So this is chromosome number 5. And XY segregated from each other 3%. So your Y can be on this side, on the left, or on the right, right? Either one of those. And then Y and Z segregated from each other 9% of the time. And then X and C is 12% of the time. So then it tells you, so if Y is here, right, and there's 3% here, right, 3 map units, and then you have, so you say this is Y, and then you say X and Z is 12%, so from X to Z here is 12 units, and then Y and Z is 9, so you add up right. Okay, but if you say, so when you do questions like this, don't get panicked. So you can try either way, because it's only, you can put Y on this side, and whatever the number adds up, that's the one you want to go with. Okay, so let's try the next one. Okay, I think so far everybody vote for C. So let's see if C is correct. So you just put X anywhere on this chromosome, doesn't matter. And you know that X and Y is 6.9. So let's start with what you guys have as C. If you put Y here, this is 6.9. Differences, map distance, and then you guys say that Z is here. And you know that Y and Z here is 7.5, right? So let's see if the numbers add up. So then the X and Z is 0.6 here, 6.9, yep. So that add up, right? You can see that. So good, C is correct. If you do the Y here, let me see if I know it. You put, let's say in the beginning, you don't know where Y is supposed to go. You don't know whether Y should go to the left of X or right of X. You can try both ways and see at the end which numbers add up to what you want to see. Okay, if you put Y over here, that means you put the 6.9 here, and then you can put the Z over on the other end. That will also work, but then you don't see the choices here that you need. Now here's another exception. So when we talk about incomplete dominance and codominance, we're talking about exceptions to Mendel's law of dominance. So Mendel was looking at black and white kind of stuff. Okay, so if you have an allele that's dominant, it's always dominating over the recessive allele. That's what Mendel is talking about. But sometimes the dominant is not black and white. It's not complete like what he's talking about, like what Mendel is talking about. Sometimes this dominant can be incomplete or co-dominant. Okay, so let's look at some of these examples. So for incomplete dominance, the perfect example is flower color. Okay, so let's look at this. Let's say the big R here will make purple pigment. And the small r, which is recessive, right? Remember, big R is dominant, small r is recessive. It doesn't make any pigment. So it ends up being white. So if you're homozygous recessive, you have white flowers. If homozygous dominant, you have purple flower, of course. But what if you have heterozygous, like this? You end up in Mendel's law of dominance. You will only see purple, correct? But in this case, in flower color, so that's what I mean that if Mendel has started with some other plants except P, he will see a very different. He won't be able to come up with the law of dominance. So you see that if it's heterozygous like this, it will be, the phenotype will be in between. It won't be purple, won't be white, it will be a lighter purple, lavender. So it's kind of like when you're little, you're mixing color. If you mix purple and white paint together, you get a light purple, right? So that explains why you can have so many different flower color, because the heterozygous always show the in-between phenotype. So that's what incomplete dominance means. That means the dominant allele is not completely dominating over the recessive allele. So in this case, you can see the recessive allele still show a little bit, right? Now here's another example. So last example was about purple. Snapdragons, the dominant allele is red. Recessive allele, once again, doesn't make any pigment, so it's always white. But if you have a heterozygous, big R, little r, it'll be pink, because when you dilute red out, it becomes pink color. So that's incomplete dominance. So we did this already, right? So if I say in a genetic question in the exam, I would say this particular allele is incomplete dominance. So when you see the heterozygous, right? When you see the heterozygous in incomplete dominance, it won't be the same phenotype as the dominant allele. It will be in between. Just remember that. Let's try this question. Okay, so this is an example of incomplete dominance. You have a homozygous recessive, too small b. You have a homozygous dominant, too big b, that's why it's black. And it tells you here too, true breeding, right? That means purebred, right? So when they have a kid, it'll be heterozygous. But remember in Mendel's law of dominance, this kid's supposed to look like the father, phenotypically. But it's not because it's incomplete dominance, so it's brown. So it's in between. The color is in between the two. So now it's asking you, what if you made the offspring that's heterozygous like this, right? So then asks you if you have 8 offspring. So you just need to multiply everything by 2. So now be very careful. So if you look at this Putnam square here, this will be white, this will be black, and these two kids will be what? Brown, right? So they're asking how many will be black. Since there's 1 out of 4 will be black, if you're looking at A, it will be 2. So you guys got it right, it's B. Right? 1 of the 4 kids is black, so they're asking how many out of 8. So it's 2. So what about codominance? Codominance means that both of the dominant allele, there's no recessive in a sense. If you have both of the dominant allele, they will both show up instead of just one of them show up. So the heterozygous, unlike the incomplete dominance, will display both of the phenotypes of the allele. So the example we're gonna use is human blood type. We know that we have A, B, O blood type, right? So let's look at this. So in human red blood cell, what you usually have a lot of times is you have something called antigen on the surface of the cell. Now, if you don't remember these antigen, actually we talk about it in the carbohydrate chapter. They're glycoprotein to identify the cells from foreign cells, right? So you can tell your cell, your body's own cell to a foreign cell that happen to enter your body. So these glycoprotein can be A. So, and if you have A kind of antigen on the red blood cell, you have A type blood. And then of course, if you have B antigen on the surface of the red blood cell here, let me change it to laser pointer. If you have B antigen, then you have B type blood. Now, so A and B are codominant. So if you have both A and B on the surface of your cell, then your phenotype is AB blood because both of them will show up. Okay. So the O type is the only one that is recessive because there's no antigen. Kind of like the white in the buffalo or the flower color. Okay. There's nothing on the surface. Okay. So this is what codominance means. If A and B both happen on the surface of the red blood cell, they are codominate. That means they both express. So let's look at another picture. So if you look at this, you know that when you get blood, A type blood usually have to get A, but here's an example. So if you have AB blood type, you can get blood from anybody, okay? So it's called universal recipient because it has both antigen A and B. So if you get A type blood, they will recognize it as A, so they won't fight against it. If you get B blood type, it's fine too because they already have B antigen, so they won't fight against B. And of course, O doesn't have any antigen on the surface, so O blood type can give blood to anybody. Just remember the antigen on the surface of the red blood cell is those identity marker. So if you don't have any identity marker, you can, O type can go into anybody without triggering an immune response. Good. The answer is C. Now, so this allele, the number of alleles in a population could be many, could be 3, could be 500, but every individual, we can only get 2, right? One from your mother and one from your father. Let's try this codominance question. Now, when you do codominance, like blood type, the way I do it is a little different than conventionally. They have a small I for O. So for this kind of question, the way I'll do it is like this. I'll show you. So it's easier, I think. So they say it's a homozygous type B. So I will just, for this one, I'll just say 2 big B. And it's mating with a heterozygous type A. Heterozygous means that if one A, this small I means O. Right? O means that there's no antigen on the surface. So half of the cell, well, no, half of the red blood cell will have the A antigen and half of the red blood cell will have nothing on it. So heterozygous look like this. So when I do a mating like this, I'll have A, O, and then 2B here. Okay, so the kids for this kid will be AB blood type. This kid will be a heterozygous B blood type because remember B is dominant. This one will be AB again. Right? This one will be a heterozygous B again. Now remember, for each A type or B type blood type, you can be AO, heterozygous, or homozygous AA. Okay, so this is the way I would do it. But if you learn blood type before and you're used to doing the I, you can do that too. But for most people, I think that's more straightforward to see. So let's try this question. Is it possible for a child to be biological to parents with A and B blood type? Ooh. Hey, people, do you think it's possible? Yep, it is possible. Remember, for anybody to have A type or B type, it can be heterozygous. Now, if the parents are both, where's my little? If the parents are both homozygous A or homozygous B, then it's not possible to have an O type. But like what In is saying, that if they are a heterozygous, then it's possible because there's an O from each parent to give the blood type O. Now let's go on. So far we've been talking about codominance and incomplete dominance. So the summary from these two is that a lot of times, just like in a real world setting, not everything is black and white. There's a lot of gray, right? So there's not a simple dominant and recessive relationship. Codominance, both of them can be dominant, and then in incomplete dominance, the dominant is not completely dominating over the recessive. And the other thing that Mendel didn't mention or didn't notice was that a lot of physical characteristic that we're looking at these days are not controlled by a single genes. And here's some example. So we already talked about multiple allelism. That means in the population, just like blood type, you have 3 different alleles, A, B, and O. But for each individual, you only get 2. The other example is human beta globin protein, and you actually did a search on the government database site for this protein. Now there are 500 different alleles in the population. But once again, each individual can only get 2 out of the 500. So you can see how many combinations that you can get, right, in a population. And if you have multiple alleles, like we talk about for a particular characteristic you're looking at, you end up with something called polymorphic traits. Polymorphic, poly means many, morphic means form. So for physical characteristic, you will see many different forms. For example, the ABO blood type, you have how many? You have 4 different phenotype or genotype, right? From 2 different alleles, and that explains why you can have that many. If in a Mendel's situation, you can only have 2, right? You have dominant or recessive, and you only have 2 alleles to look at, so it's a lot simpler. What time is it? Okay, let's take a break for 5 minutes. When we come back, I'm going to talk about human pedigrees, and then we'll talk about the lab and see if you have questions about the worksheet. Let's take a break for 5 minutes. Okay, let's look at some pedigrees. And I think you have to do pedigrees in today's lab. Um, well, because it's not legal to do human experiments, so a lot of human genetics are done by looking at family trees, which is also called pedigrees. I think they call pedigrees in animals, in dogs a lot. So here's an example of pedigrees. Use a pointer. You see that male is always represented by square, female is always in circle. And a lot of times, the individual that has the whatever disease they're looking at is always shaded like this. Now, they show that there's half shaded and half open. A lot of times, and this represent carrier, so it's heterozygous, right? But a lot of times you don't really know that. So if they phenotypically appear normal, they just do a little white circle because a lot of times you can't really tell. And just like family trees here, what you're looking at is you have your dad and mom here on top, and then they have kids, right, going up here. This is a boy that they have, and then they have a girl, and then they have another girl, and this girl get married to another boy here. So that's how you read it, okay? Now, for human genetic disorder, usually is because of defective alleles or recessive alleles. So most genetic disorder is autosomal recessive. That means they occur in the first 22 pairs of chromosomes, and they are autosomal recessive. That means that they have to be homozygous recessive in order to show the disease, okay? Heterozygous, they are called carrier, and they will appear normal. Only homozygous with two defective alleles. Remember, we're looking at the autosomal, so it's the first 22 pairs of chromosomes. So only have homozygous recessive will be having the disease, and that is a good thing because it's very hard to have two defective alleles come together like this, right? So the chances of having genetic disease in the population is really low. Now, here's an example of autosomal recessive disease. It's called sickle cell anemia, and we use the same example over and over again. In the protein chapter in the beginning of the semester, we look at sickle cell. Remember, that's the one. The cell becomes sickle shape, which is half a donut, right? And it is autosomal recessive. That means you have to have homozygous, both of the recessive allele, to show this disease. Now, this is the same thing we talk about in the beginning of the semester. Is a single nucleotide change mutation cause the shape of the protein to change, because remember, the nucleotide sequence will determine the amino acid sequence in the protein, which will then determine the protein shape and structures. Now, sometimes in the case of sickle cell, it is more prominent. So if you look at genetic disorder, sometimes it's more prominent in certain area. And there's a reason why. For example, sickle cell here, I have a map of Africa here. Now, it turned out that the carriers for sickle cell, which is heterozygotes, they are resistant to malaria. So these individuals get to survive in Africa better. Okay, so if you look at this map here, where is it? Sickle cell. So the blue, so the sickle, what this two map is trying to show you, the percent of sickle cell individual matches the area of malaria, because sickle cell in the heterozygous sickle cell, which is physically normal but carry a recessive gene, is actually resistant to malaria, so then they can survive better in this region. But the problem is, if heterozygotes keep having babies, then they're more homozygous recessive individual in this region too, okay? And that explains why you have more sickle cell incidence in the area where malaria is very prominent. Make sense? So sometimes the environment factor will select out certain genetic makeup that fit the environment better. Just like what we talked about in chapter 1, remember? If they survive, they have babies, and then you can keep seeing the same kind of genotype over and over again. Now, we just talked about autosomal recessive. Remember, autosomal recessive, the word autosome means that you have to be the first 22 pairs of chromosomes, those alleles, right? And it has to be recessive, so they have to be homozygous recessive. Both alleles has to be bad. Now, what if it's autosomal dominant? If the disease is autosomal dominant, it's like what? Huntington disease, okay? So that means you only need one copy of the dominant allele for the individual to show the disease. So there's no carrier in this scenario, okay? The carrier, so the heterozygous and the homozygous dominant individual will both have the disease, okay? So here's an example of an autosomal dominant pedigree. Now you can tell that the female has a disease and they cannot be carrier, so they have to show it. So then it's filled here, and then the normal man here, they have kids. Can you see that all the kids, 1, 2, 3, they all have it? Because let me change the pen. So for this female, autosomal dominant, let's say this is Huntington disease, right? She could be big H, heterozygous, little h, or big H to show the symptoms, right? All it needs is one dominant allele. For the normal individual like this, he can only be 2 small h, because if he has any big one, he's going to show, right? Now, based on this pedigree here, you see that all 3 kids have it. So the probability of the female having this genotype is higher, because you can see that big H go with one of the small h. So all the kids here could be heterozygous. Can you see that? And they will all show it. Now, if the female were this, and then the male is this, right? Then it will be 3 to 1. But we only have 3 kids here. Maybe the last one will be normal, so we don't know that. But this is just to show you that, but you can kind of, if you look at this, so for this individual here, this guy has to be normal, right? This woman has to be normal. So for any normal person, they will have homozygous recessive, right? For any person with a disease has to be heterozygous or homozygous. If you look at their kids, you can kind of guess what this person is. This one has it, but this one is normal. Look at this. So that means this person here has to be this, which is good, right? We know that she has to be this anyway. But anyway, so this is how you're gonna look at it. So for autosomal dominant pedigree, it always show you one of the parents will have the disease, and then most of the kids will have it. Okay, remember, even though it's not like in this case 100%, it's always 3 to 1. So 75% of the kids will have the disease, and one of them will be normal. That's how you're gonna tell. So let's try this. Now, before you do this one, let's talk about this. So how do you know whether it's sex-linked, right? So to see whether it's sex-linked, all you have to do is look at the kids. If both the male and the female get the disease equally, then it's not going to be sex-linked. If it's sex-linked, it will tell you that mostly boys gets it or mostly girls gets it, okay? So that will be the key thing. But in this case here, if you only see one boy and one girl and they both have it, that means 50-50% chance. So this is not going to be sex-linked, okay? So what do you think this one is? Yep, the answer is A. So let's see. So for the parents, this could be big. You know that from this, the parents has to be heterozygous. So they will appear normal. Dang it. Right? So the two small a's will come together in kids. That's why they have the disease, right? Okay, let's look at this one then. Now the keyword here is in a recessive manner, right? The keyword here is definitely. Okay, so some of you vote for D. So let's see, here's the key, right? It tells you too small d is that. So we know that this individual has to be too small d, right? Let's do the one we know for sure. This 2 has to be too small d, right? Now, these are normal parents but have a deaf child. That means they have to be carrying the deaf genes. So these parent has to be big D and small d, heterozygous. Agree? So then the two small d from each parents, the small d from each parents will go and combine, become the kid here, right? Now, but when this kid marry, they also have a deaf kid. What does that mean? That means this wife here has to also carry a recessive gene. So she'll be a heterozygous. So then one of the small d from the father, one of the small d from mother, and become a deaf child. Yeah, so the answer is D. This individual, we don't know. This could be two big D together, right? Or big D and a small d. See that? So this one we don't know for sure. But these we know for sure. So it's 1 and 2, the mom and dad, and then number 3 here. So answer is D. Good job. So we're done with today's human genetics. So what I want to do, give you a minute to have questions. If not, I'm going to pull up the lab first. Lab. Where is lab? Here. So you're supposed to have done part 1 already. So today we're supposed to do part 2. Okay. And we are not doing, are we all there? On part 2 of the lab, we are not doing PTC tasting. Okay, so because that's what you need to, there's a piece of paper with PTC and you're supposed to taste it in the lab. So we are not in real lab right now. So you're not going to taste it. So just cross that one out. But you can do tongue rolling. So what you're going to do is you just look at yourself. Can you roll your tongue? If you can roll your tongue on the phenotype here, you just say tongue roller or non-tongue roller, something like that, a descriptive word here. Genotype here, on the other hand, we're going to talk about that. So why don't you guys go down the row and do this? And you know what widow's peak look like, right? The little pointy peak along your hairline. I pull up pictures for hitchhiker's thumb. Can you see that? It kind of look like this. That your thumb can bend backward really by a 90-degree angle, like these guys here. Can you see it? So this one, let me blow it up. This is a good example here, showing you this is a normal thumb. This is a hitchhiker thumb. You can bend it backward a lot more. Okay, and then attach earlobe. And here's an example of attached earlobe. Okay, when the earlobe is completely attached to your face versus is free hanging here, that's not attached. Can you see that? So you kind of look at your own earlobe, see whether they're attached or not. So that's what you're going to write here. Widow's peak, you say yes peak, no peak. Hitchhiker's thumb, yes or no. Attached earlobes, yes and no. And then for the genotype part, you'll be able to do that from the description here. In the front, this part here on page 2 on the genetics lab. Now, because it tells you here, tongue rolling is dominant. Can you see that? So if you're able to roll your tongue into a U shape, you have a dominant gene. But that could be homozygous dominant or heterozygous dominant. So if you can roll your tongue on your genotype, you could be 2 big R or big R and a small r. But if you cannot roll your tongue, kind of like me, you have to be 2 small r. Does that make sense? So that's what you're supposed to put on the genotype part here. 2 letters, uppercase, lowercase, whatever it is. So if you look at widow's peak, you do the same thing. If you look at widow's peak, it tells you widow's peak is, once again, controlled by a dominant gene, right? So you just follow the same logic as what I talk about in tongue rolling. If you have it, then you have to be heterozygous or homozygous. If you don't have it, you have to be a recessive, homozygous recessive. Now here, hitchhiker's thumb is different. It's by recessive gene. So if you have hitchhiker's thumb, then you have to be homozygous recessive. If you don't have it, then it's either heterozygous or homozygous dominant. Right? Attached earlobe, what is it? It's recessive again. They even give you the letter to use. So just follow what they have right there. Are we clear how to do this table? I think this is the only table I want to talk about. And then the next one here. Any questions first? So I'm going to give you a few minutes to write that down. And then I want to talk about question number 7. We're going to talk about a couple of them, just to be sure. And it's U shape, not W. Some people can do the W one, which is kind of scary, but when you can roll your tongue, it's like U shape, just like what the description is saying, not W. Okay, so let's look at this. So for question number 7 here, they're saying that if a person is homozygous for tasting PTC. Okay, let's look at what PTC is first, since we're not doing it. So PTC is the ability to taste. So PTC is a chemical. So if you can taste PTC and it's bitter. So if you can taste PTC, you have a dominant gene. Okay, so it's a big T. So people that can taste PTC is either heterozygous dominant or homozygous dominant. So people that cannot taste PTC is going to be homozygous recessive. Now, even though we don't have the PTC paper except Misty, if for those of you that really can taste the bitterness in vegetables, in coffee, in chocolate, you are probably PTC positive, okay? Because you're very sensitive to bitter taste. For those of you that cannot taste any bitterness in the kind of stuff I talk about, you're probably not a taster. Okay, so for a PTC taster, you need to be homozygous dominant or heterozygous dominant because it's dominant. Okay, so now this question is talking about What kind of gametes will you have if you were, so if a person is homozygous to taste PTC, that means it's too big T, right? Her genotype, I just gave it to you. Her genotype will be too big T here, homozygous. What about the genotype for that person's gametes? So if all he has is too big T, then the gametes is 100% big T, right? You only have one T instead of two. So the genotype is a somatic, showing you two alleles. But in gametes, you only have one of those alleles. But since he is homozygous, so 100% of the gametes will be big T. You getting it? Yes, Misty, if you know you can taste it, you can go ahead and do that row about PTC. Now, here's another more trickier question. What if that person is heterozygous? So the genotype will be big T, small t, right? Heterozygous, still be able to taste. But that person's gametes will be what? The big T and small t will separate during meiosis. So the gametes will be 50% big T, 50% small t. Agree? Now, so I show you the first two. You should be able to do C. It's talking about unable to taste, right? So it has to be homozygous recessive. And then you can do this one too. You can do a little Putnam square showing now Jane is a non-taster, right? Husband is a taster, but the hint is his mother is not. So that tells you what kind of taster he is. Okay. Now, so you can cross Jane and husband Joe on a Putnam square and see what the chance of their kid being a taster, right? And remember, you have to show the genotype of both parents and here. So the last question here is about pedigree. And they give you all the information. So when you do this pedigree, I think you can do, remember the symbol, square means man, circle means female, right? If you know it's a carrier, you can do half shaded or not. Now, but for each symbol here, you have to write the name next to it. So let's say if you start with the husband, Alan, and then you have the square, you have to label Alan next to the square. And you also have to, besides the name, you also have to write down the genotype for each of the symbol. Is it too big T, too small t, a heterozygous big T, small t? You have to do all of it next to each symbol, okay? Besides shaded, square, or circle. Get it? So that's what I want for this pedigree. You need to be very, very clear. And then the bottom, they ask you for genotype. If you label on top, then you have no trouble just copying down here. Some of the genotype is not sure, so you can do either or. So that person can be either homozygous or heterozygous taster, right? So that's what what I expect you to do here. Now for the worksheet, what I want to talk about, if you have your worksheet, I want to talk about question number 4 with the pedigree. A lot of you get this one wrong. So if you pay attention to this pedigree here, you see that the parents here, one of them already has the disease. And if you look at like the kids, right? 1, 2, 3, 4, 5. 3 out of the 5 kids had the disease. Okay. Now, so remember I showed you an example. If one of the parents has it, most of the kids has it. So this has to be what? Autosomal dominant. Right? So just remember that. So this is not going to be recessive because you're already showing it to you. Okay. And then if this is autosomal dominant, then you have to, so then will be this, right? Then based on this, you'll be able to figure out the genotype for each of these individuals. And I think you'll be okay with the rest. And the last question on the worksheet is doing a pedigree too. Make sure you follow all these examples here I have about big H, little h. You label each of the symbol with the name of the individual and the genotype of the individual. Okay, so this is what you're supposed to do. You're supposed to draw a pedigree, label each person, right? So that's all I have for today. If you have questions, please show me now. Look at here first. No quát cả. Đi làm mồi quát cơ mà. Mình trong này với mình. Mình mồi. Okay, let's do Misty's first. So in autosomal dominant, this example here, I gave one exactly like that in the PowerPoint. Let's look at it real quick. This one, this is the one on the worksheet is exactly like this one on the PowerPoint, okay? So what I was trying to tell you is that the parents usually have one that already showed the disease and most of the kids will have it. So if you look at this worksheet here, one of the parents will have the disease, right? And 3 of the 5 kids, 1, 2, 3, 4, 5, has the disease. So that's why I say for number 4, it's going to be autosomal dominant. Okay. Part 1 of the lab, how would the Putnam square for Q2 look like? Let's see, part 1 of the lab. Part 1. Part 1 of the lab. How would the Putnam square for question 2 look like? Oh, good question. So someone else asked that. Remember for each penny, you have either H or T, head or tail. So it's saying that for the gamete, it will be either big H or big T here. And same thing here, big H or big T here. So that's how you do your crosses, okay? So for Penny, it says that 2 possible alleles. That's what it means. So for this individual, it's heterozygous, right? So it's big H and big T. So then you have one big H here and one big T here, one big H here and one big T here. Now, and that also helps you to answer B here. Does this situation, if you answer codominance or incomplete dominance, is fine. If you can explain your way out, but for me, it looks like it's codominance because both of those are big T, big letters, big H or big T, right? Right. So Misty, for autosomal recessive, what happened to the pedigree is that both of the parents will look normal, just like in this one here. Just like this one, both of the parents will be heterozygous a lot of times, and they will look normal. Okay, and then you will see that only one of the 4 kids will have it because it's autosomal recessive. You have to have homozygous recessive to show. But in autosomal dominant, in this case, one of the 2 parents will have it, and most of the kids will have it because it's not 1 to 3 again anymore. It's the 3 this time is on the disease side. So the 3 out of the 4 kids will show the disease. Okay. Basically, the bottom line, Misty, is to remember for autosomal dominant disease, um, a lot more children will be affected by the parents, by the diseased parents. Okay, because you only need one dominant gene to show the disease, versus in autosomal recessive, you need 2 recessive genes. So the likelihood of getting the disease is a lot lower. So you get 1 out of 4. So the way to think about it is 1 out of 4 will get disease in autosomal recessive, but in autosomal dominant, it's 3 out of 4 will get it. Any other questions before our times run out? Okay, one thing I forgot to say is on exam 5, this is all the material we're going to cover in exam 5. So exam 5, we have replication, DNA replication, we have meiosis, and then we also have genetics. Okay, it'll be the same format, open book, open notes on D2L. And then next chapter, which is metabolism and cellular respiration, is the last chapter of the semester, but it's also the most difficult chapter of the semester. So you guys need to make sure you do your homework and worksheet and stuff like that. Okay, and then we'll start on the biofuels project on what is it? Next Wednesday. I'm going to talk about it, and then so you guys know what to do. And biofuels is the last lab there is in the semester too. Yes, Misty, very good. Just like the wrinkle, seeds skip a generation to appear, right? When you look at the wrinkle seed, the parents has it, and then F1 generation don't have it because everybody is heterozygous. And then the F2 generation, when you have the 1 to 3 ratio, you start seeing the wrinkle as the one out of the four. So you're very good. So that's what happened. If you're autosomal recessive, that's what happened. So I will open up exam 5 maybe around Monday or Wednesday next week, and you will have until, let's say, you have a week to finish it. Okay, so you have time. So I open up exam 5 maybe next Monday. If I open it up next Monday, it will be due the following Monday. If I open up the next Wednesday, it will be due the following Wednesday. Any other questions? We have 3 minutes left. Okay, so Misty is saying on the lab for the class data, no, you don't do class data. So for this part here, you cross it out because we don't have a class. So you just do your own penny tossing and then just get your own data here. Part 1, number 3. Yeah, right. So you just don't do the class data, just do your own lab group, which is you and yourself. Okay, good. So I will see you guys again on Monday. Bye.